And thank you for this opportunity to be able to present to all of you. I'm really excited to be able to tell you about how we're using organoids to elucidate mechanisms of neuroendocrine cell growth and tumorigenesis. So to begin, I'll tell you about the tumor type that I'm most interested in, and these are neuroendocrine neoplasms. These are a heterogeneous group of rare tumors that are thought to arise from the cells of the diffuse neuroendocrine system. And what that means is that they're thought to arise from a very rare cell type called the neuroendocrine cell. Now, the most famous neuroendocrine cell is the pancreatic beta cell, and that is the cell in the pancreas that's responsible for releasing insulin in response to glucose. But there are actually a lot of different neuroendocrine cells, including other neuroendocrine cells in the pancreas, as well as neuroendocrine cells in the lung and the gastrointestinal system, for example. What this means is that neuroendocrine neoplasms, or NENs, can arise in any number of different epithelial tissues, basically any epithelial tissue that has neuroendocrine cells, which is most epithelial tissues, can eventually be the site for a neuroendocrine neoplasm. Now, what you can see in this diagram are the most common sites for neuroendocrine neoplasms, and so those are the lung and the gastroenteropancreatic system, and those are the NENs that I'll be focusing on mostly for this talk. What I've done here is I've outlined some of the challenges that NEN researchers face when trying to study these tumors. The first is that NENs are actually comprised of two different, or what are thought of as two different entities. And those are neuroendocrine tumors, or NETs, which are low-grade tumors, and then neuroendocrine carcinomas, or NECs, which are actually very high-grade tumors. And NETs progress and proliferate very slowly, but NECs are very highly proliferative, highly aggressive disease that's associated with a very poor prognosis. And currently, we don't understand whether or not there's a relationship between NETs and NECs beyond simply their shared expression of neuroendocrine markers. Despite the fact that many NENs are these low-grade NETs that I was talking about, and so they may follow a relatively more indolent course compared to other tumor types, NENs are still a very significant clinical problem. That's because the majority of patients are actually diagnosed at advanced stages. And there are very limited treatment options for these tumors, and we don't currently understand how to predict which patients will respond to the treatments that are available. These tumors are also relatively rare, which means that we have relatively fewer patient samples to learn from. And finally, one of the biggest challenges that we are faced with is the fact that there are very few preclinical models that can be used for the kinds of mechanistic and drug response studies that are required in order to try to identify new therapeutic avenues for these patients. And so the question that I asked when I joined the lab was, can we use organoids as a tool to study NEN biology? And to begin, I'll just give a very brief introduction into organoids. And because it was the first organoid system established in the lab, I'll be using the small intestine as an example. So in general, adult stem cell derived organoids, using this organoid technology, we can isolate a single adult stem cell, in this case from the intestine, and then grow it under defined conditions such that over the course of 2 weeks, as you can see from these different snapshots at different days, this single cell will proliferate, generate new cells. These new cells will differentiate, and then all of these cells will self-organize to form a structure that resembles the structure of the organ from which this cell was originally derived, in this case, the small intestine. Now, if we look at this day 13 image, what we can see is that we have this complex structure where we have protrusions and then these inner parts. And this really recapitulates the structure of the intestine, where the intestine contains a stem cell compartment called the crypt, and then a villus, which contains all the differentiated cell types. What we find when we analyze this organoid structure is that all the stem cells are contained in these protrusions here, so these are the crypt regions, and then as we go further down in here, we see that these represent the villus regions and contain all the same differentiated cell types that are found in vivo. Now, an important feature of this organoid technology, and maybe it's something we don't talk a lot about, is the fact that we use a defined media. And so what that means is that we try to avoid using things like fetal bovine serum, and instead try to add the specific factors that are required for the cells that we're interested in growing, for them to grow. And so usually the way that we do this is we design a media based on what is known about the niche microenvironment in vivo. And so what I'm showing you here in this diagram on the left is that in vivo, in the small intestine, this crypt region, we know that the niche microenvironment presents cells with things like BMP inhibitors, Wnt signaling molecules such as Wnt ligands and R-spondins, as well as EGF ligands. And so what we did is we used that information to design a media that contains each of these factors. Indeed, we find that each of these factors is important for promoting the growth of these stem cells in vitro as organoids. Importantly, what this means is that we can modify this media and really adapt it to specific cell types, and these modifications and adaptations have allowed us to now expand this technology and given us the ability to grow organoids from a large array of different kinds of adult tissues, including human lungs, stomach, colon, liver, pancreas, and many other tissues. And in general, if we can grow the normal tissue, we can also grow diseased tissue. And in these cases, often what we find is that the diseased tissue organoids can serve as surrogates for the patients from which they were derived. So in the years following the establishment of the first organoid, really we have expanded much further, and this system has really proven to be a very powerful system for the study of stem cell biology and disease. And the one thing I want to highlight is that this system is unique in that it allows us to study both normal cells and disease cells in vitro. And so this really gives us the unique opportunity to try to understand normal cells and how those normal cells can become, for example, tumor cells. So the stories that I'll be telling you about today really take advantage of this ability to look at both normal cells and tumor cells in culture. In the first half of the talk, I'll be telling you about our efforts to study NEN tumor cells by building a NEN patient-derived organoid biobank of intestinal, lung, and pancreatic NECs and NETs. And then in the second half of the talk, I'll try to tell you about how we're using normal organoids to study normal neuroendocrine cells of the lung or pulmonary neuroendocrine cells. So in order to study NEN tumor cells, in collaboration with a number of different clinical institutes in the Netherlands, we've set out to try to generate a biobank of patient-derived organoids or PDOs from NEN tissue. And the idea is that once we've established organoids from these tissues, they will be hopefully representative of the tissue they were derived from, and then we can use these organoids to do a large number of studies, including drug screening to try to identify therapeutic vulnerabilities, as well as genomic analyses, and of course, hypothesis-driven experiments to look at specific gene function, for example, using CRISPR-Cas9. So what I'm showing you here is what we've done to date. Since January of 2017, we've been isolating samples we've isolated close to 200 NEN tumors and metastasis samples. And what I'm showing you here is a summary of our success rate for the PDO derivation from each of these samples. And what you can see is that we have close to 100 NEN PDOs currently growing in culture. And what you can see on the bottom is I've summarized our estimated success rate. Now I say this is an estimated success rate, and I put down a range because, as I mentioned, the nets are very slow growing, and that's true both in vivo and in vitro. So the organoids are also slow growing. So for some of these, it's not clear whether or not they will stand the test of time. But at the moment, this is our estimated success rate. And what you can see is that it really varies from tissue to tissue. And so what we think is that perhaps there are some modifications that need to be made to the media in order to specify it to each tissue. What you can see on the bottom are some representative images of what these organoids look like. You can see that they don't form these cystic structures that maybe one would be more used to looking at, but instead form these sort of dense structures that sometimes form branching structures. Here I've summarized what our biobank currently looks like. On the left, what you can see is the clinical distribution of NENs, and this really highlights what I told you in the beginning of the talk, which is that the majority of NENs are found in either the lung or the gastroenteric pancreatic system. Here on the right, I'm showing you the tissue distribution of the PDOs in our NEN biobank, and I really want to highlight that we've successfully generated 41 NEN PDOs. This is really exciting for us, and I think for the field, because currently there are very few in vitro model systems. There's only a handful of cell lines for this tumor type, so this has been really exciting to generate. And this NEN biobank is representative of what the most common types of NENs are in vivo. So we have the lung and the gastroenteropancreatic system represented. And then we also have some more rare subtypes, including Merkel cell carcinoma and a neuroendocrine carcinoma from an unknown primary. So before I move on, I want to just pause and take a second to think about one of the things that I mentioned in the beginning, and that is that we use a defined growth media, and that we design that media in order to recreate the in vivo niche. So what that means is usually we're going from the information that we have from the in vivo environment and then using that and applying it to the in vitro environment that we're trying to recreate. The problem is that in the case of NENs, we really didn't have a lot of information about what the microenvironment is composed of, and perhaps there was some information out there, but it wasn't clear what was important and what wasn't important for the growth of these cells. And so what we found is that actually generating this media was an experiment in and of itself that I think has highlighted or given us some insight into the biology of this disease. And so what we found is that some of the components of the media that worked are components that are found in many different organoid media. These include R-spondin and BMP inhibitors, but there's some other components that were more specific to NENs in particular. And so I wonder whether or not what we can do now is ask whether or not what we're learning here in the in vitro environment is telling us something about the in vivo environment. And I think this highlights 2 points of reflection that I want to point out. So the first is that this media optimization, perhaps if what we find is important for in vitro growth is also important for in vivo growth, and this will have to be tested, but if it is, then it perhaps is telling us something about potential therapeutic vulnerabilities for these tumors. And then the other thing is something that I don't really have any data to speak to, but I think is something to think about, is the fact that this media was optimized using our NEC PDOs. These are the high-grade PDOs, and that's because they were easier to work with and proliferate much faster. We had more material. But what we find is that what worked for the NECs also works for the NETs. And so this raises the question whether or not this implies that NETs and NECs have similar niche factor dependencies, despite the fact that one is a low-grade subtype and the other one is a high-grade subtype. So one of the most interesting observations that we made when we were developing this media and figuring out what worked and what didn't was the fact that one of the things that we added to the media, this metabolite, is a metabolite that we normally add to organoid medias in general. It seemed to actually inhibit the growth of these tumors. And so what I'm showing you here are some pictures from one of the experiments where we made this observation. So what you can see on the top is that when we grow normal pancreas organoids in the absence of this metabolite, there's essentially no outgrowth. As we add increasing concentrations of this metabolite, we see that there's increased outgrowth, and actually this is the concentration at which we normally grow these organoids. However, if we look on the bottom at these examples of a pancreas neck and a lung neck organoid, what we can see is the exact opposite pattern. So now the best outgrowth is in the absence of this metabolite, and as we increase the concentration of the metabolite, we see decreased outgrowth. And so we think this might be telling us something, pointing us towards a potential therapeutic vulnerability. This is something that we're looking further into. We have extended it to other NEN lines and other NEN subtypes. It seems to be consistent, and so at the moment we're trying to better understand what's happening and whether or not it can be exploited therapeutically. Naturally, we're also performing molecular analyses on these PDOs. And of course, one of the first things we wanted to know was whether or not these PDOs actually express the neuroendocrine markers that are so characteristic of the primary tumors. And so what I'm showing you here is some qPCR data from some lung NEN PDOs and intestinal NEN PDOs. And what you can see is that indeed these PDOs all express significant levels of two canonical neuroendocrine markers, chromogranin A and NeuroD1. We've also done RNA sequencing on these PDOs, and we do find that the global expression patterns are consistent with primary tumors, but I don't have time to tell you about that. We have also gone further into this and are trying to do a large-scale omic analysis of matched PDOs and primary tumors. And to do this, we've teamed up with a group spearheaded by Drs. Linet Fernandez-Cuesta and Dr. Matthew Foll at the International Agency for Research on Cancer. And through their pan-nenomics effort, they're essentially performing in-depth omic analysis of a large number of primary NENs from many different tissue sites from around the world. And what they've done is they've applied their bioinformatic pipeline to analysis of data from our PDOs and primary tumors. And so I don't have a lot of time to go into all of the data, and we've only just started getting it back, but I wanted to highlight one of the interesting things or most exciting things that we've been able to do with the analysis of the data that we have. So using whole genome sequencing analysis, we can compare the primary tumor to the PDO that it was derived from, and then using the frequency at which a specific small variant allele is found, we can look at the intratumor heterogeneity of both the tumor and the organoid. And so what I'm showing you on this graph are essentially each dot on this graph, it represents a small variant, so a mutation. And where it lies on the graph tells you the frequency at which it is found. So essentially, this P53 mutation that I've highlighted here lies on the top right-hand corner. So this is found in pretty much all of the cells, whereas this mutation here, for example, lies lower down towards the lower left-hand corner. And so it's found in a smaller fraction of the cells. Now, the important thing I want to highlight is the fact that the majority of these dots all lie along this diagonal line. So that tells us that most of the mutations that are found in the primary tumor are not only also found in the PDO, they're found at pretty much the same frequency at which they're found in the primary tumor, suggesting that despite the fact that this organoid spent almost a year in culture before we performed the whole genome sequencing analysis, it was able to maintain the genetic diversity of the primary tumor at more or less the same rate. Another thing we can do using this information is start to recreate phylogenetic trees, and this allows us to infer subclones within the tumors and the PDOs. And what I'm showing you here is the phylogenetic tree that was generated using this data for this particular sample, and what we can see is that we can infer about 5 subclones. If we then look at the tumor and the PDO, what we can see is that most of the subclones that are in the tumor are also found in the PDO. This to us is really exciting because it really shows that the PDOs are able to maintain the intratumor heterogeneity of the disease. We do see that there are a couple of subclones that seem to be changing in frequency, and this is something that we're delving further into to try to better understand. But I think it's really interesting to see that we are able to maintain different subclones within these cultures. So just to summarize this first part of the talk, what I've shown you is that we have generated a NEN PDO biobank, which consists of 41 PDOs derived from high-grade and low-grade NENs. And these include rare subtypes such as neuroendocrine carcinomas from an unknown primary and Merkel cell carcinoma, which is a NEN of the skin. We find that our NEN PDOs express canonical neuroendocrine markers and show global expression patterns consistent with their matched primary tumors, and NEN PDOs preserve the genetic alterations from the primary tumor, including the intratumor heterogeneity of the primary tumor. So in the second part of the talk, I'll tell you about how we're trying to use this system to also understand the normal neuroendocrine cells. And so the idea is that if we can learn more about normal neuroendocrine cells, we can perhaps understand why certain signals might be driving the growth of tumor cells, for example. So in the lab, prior to me joining, a previous postdoc in the lab established airway organoid, what we call airway organoids. So these are organoids derived from human lung tissue. And what I'm showing you here is some of the characterization that Norman did on these organoids, and using this characterization, through this characterization, he was able to show that these organoids express keratin 5, so they have basal cells, they express markers for club cells, which is a secretory cell type. You can see here a nice electron micrograph of cilia, so they contain ciliated cells. There's also staining here, as well as goblet cells with which secrete mucus. And so based on this cell type composition, we call these organoids not lung organoids, but airway organoids. That's because the cell types that we find are consistent with the airway. We don't find alveolar cells, for example. Those are found in the terminal airways, and so they're not alveolar organoids. However, if we look at these cell types and then look at the cell types that we're enriching for, we know we're enriching for basal cells, which I've shown you here. These are the putative adult stem cell in the lung. However, the cell type that I'm most interested in, the PNECs, which are the pulmonary neuroendocrine cells, are not found. And so in order to try to get around this problem, when I joined the lab, I decided to try to generate airway organoids from fetal lung tissue. And the idea here is that because fetal lung tissue contains a higher number of pulmonary neuroendocrine cells than adult tissue, I thought I might be more likely to find success in culturing these cells. And then the other thing I did is I decided to look at different time points of these organoids in culture to see what was happening to these cells over time. And so what I'm showing you here is qPCR data from 3 different organoid lines derived from fetal airway tissue at an early passage, passage 2, and we're looking at a marker for basal cells, keratin 5, and a marker for neuroendocrine cells, ASC1. So what we can see is that both of these, the markers for both of these cell types are expressed at this early time point. However, if we now look at these same lines at a later time point, what we can see is that indeed we seem to be enriching for the basal cell marker, but we've essentially completely lost expression of the PNEC marker, suggesting that we're losing these cells over time. And so looking at this data, I came up with two potential explanations. The first is that PNECs are terminally differentiated cells, and so our standard airway organoid conditions maybe just don't promote the differentiation of the putative adult stem cell basal cells to PNECs. The other possibility is that there's a different progenitor cell for PNECs, and that this progenitor cell is not present in our human fetal airway organoids grown in standard media. And so in order to try to get around this and solve this problem, what I did is I decided to isolate organoids from fetal lung tissue again, but this time, again, it's all about the media. I put some of the cells that I isolated in standard airway organoid media, and I continued that long term. And then I put another group of cells from the same donor always in different media compositions, trying to figure out whether or not one of them would allow me to maintain the presence of PNECs in the cultures. I don't have time to go into everything that I've tried, but I did indeed find a media condition that allowed me to maintain PNECs in these organoids long-term, and I call this PNEC expansion. What I'm showing you here are some IF images, whole mount images of airway organoids from the same donor grown in either standard airway organoid media or in my PNEC expansion media. And what you can see is in the control, there's no cells that stain positive for this neuroendocrine marker UCHL1, but on the right, you can see these organoids grown in my PNEC expansion media do indeed contain these PNECs, and I see this that they can be maintained long-term, multiple passages. Maybe the latest passage I've looked at is passage 30, we still see PNECs. So based on this, I then said, okay, we have an expansion media. Can we now use this to further enrich for PNECs in fetal airway organoids by promoting further differentiation towards the PNEC phase? So essentially we have an expansion media, can we design a differentiation media? And so that's what I set out to do, and indeed I was able to come up with what I call this PNEC cocktail. So it's a cocktail of small molecules that appear to promote the differentiation of PNECs. And so what you can see here is that if we look again at three different fetal airway organoid lines, we're looking at a basal cell marker, TP63 this time, and then a PNEC marker. What we can see is that when I add my PNEC cocktail, we see some changes in the expression of the basal marker, but we see very dramatic increases in the expression of our PNEC marker ASCL1, suggesting that we really are increasing the number of PNECs found in these organoids. And actually, if we do IF staining again for this UCHL1 neuroendocrine marker, again, we can see that whereas before we saw, maybe a single PNEC here and there in the organoid, now we see large clusters of PNECs in our organoids, really suggesting that we have been able to identify the conditions that promote differentiation of the progenitor cell towards the PNEC cell fate. And so here I'm just summarizing what I've shown you. What you can see is that we start with standard airway organoid media. We get a composition of cells that's enriched for basal cells and has some other cell types that are found in the airways. If we instead use PNEC expansion media, we can also get basal cells, some club cells, and then some PNECs, and then we can further promote the differentiation towards the PNEC cell fate by adding a PNEC cocktail. Now, if we assume that the putative adult stem cell in the lung is indeed the basal cell and that basal cells give rise to PNECs, then this should be possible. So I should be able to start from my standard airway organoid media, add my PNEC cocktail, and get this. And perhaps it's just that I didn't have the right conditions before, and these differentiation media is the answer. So I went back to these lines, and I wanted to compare organoids derived from the same donor that had either been isolated and maintained in standard airway organoid media or in my PNEC expansion media, did the same experiment, added the PNEC differentiation cocktail, and what we see is indeed what I had expected, which is that as we saw before, if we start with PNEC expansion media, we see a dramatic increase in the expression of the PNEC marker when we add our PNEC cocktail, but we see no change when we add the PNEC cocktail to organoids that were isolated and maintained in standard airway organoid media. So essentially, this is what we see. And so what I suspect is that, or the hypothesis that I've come up with based on this data, is that perhaps our fetal airway organoids that were isolated and maintained in PNEC expansion media contain a population of progenitor cells that can differentiate to PNECs, and that this progenitor cell is not present in the fetal airway organoids maintained in our standard media. In order to try to get at the bottom of this and determine whether or not this is the case, we're performing single-cell sequencing, RNA sequencing of fetal airway organoids in our standard media, in our PNEC expansion media, and then at different time points after adding our differentiation cocktail. Now, I don't have all of the data back yet, but I do have the data just from the standard media and the PNEC expansion media. And so I'll give everyone a teaser into what we found so far. What we found so far is looking at the analysis of this data. So each dot on this TSNE plot represents a single cell, and where that single cell lies on the graph tells you how it related is to the other cells next to it in terms of its global expression patterns. And so based on this, they form different clusters. And then if we look at what's expressed in the cells in each of these clusters, we can see that we have several clusters of cells that express markers of basal cells. We have club cells. We do find our PNECs here. We have some other specialized cell types such as tuft cells. And then we also have some more progenitor-like cells here. Now, if we then look at this data with just looking at which condition the cells came from, we can see that most of the basal cells come from either our standard airway organoid media or our PNEC expansion media, but really these progenitor cell-like clusters seem to be coming only from the PNEC expansion media. And as we saw before, the PNECs are only coming from the PNEC expansion media. So we have some potential suspects for what might be giving rise to the PNECs, but we are still waiting for the data from our differentiation time course to try to really delineate this time point. Now, there is one more thing that we have been able to do with this data, and of course, whenever you're working with organoids, what you hope is that the cells that you're looking at in vitro are actually representative of the cells that are present in vivo. And so with the help of Jose Luis McFarland from Cole Trapnell's lab, we've been able to incorporate our data from single-cell RNA sequencing of the organoids with data that was provided to us by Alyssa Miller and Jason Spence's lab. From primary fetal tissue, lung tissue. And so doing this, and of course looking at the expression patterns of each of these cells, we see different clusters of cells. And if we look at where these clusters of cells, where the cells that are in each of these clusters are coming from, what we can see is that indeed our organoids the cells from the organoids are clustering together with the cells from the primary tissue, which is very comforting. It shows that there is actually congruence between the primary fetal tissue and the airway organoids, the cells from the airway organoids. And then I also want to highlight that indeed the organoid cells are contributing to all the clusters except for 4. And these 4 clusters are actually, we didn't expect to see organoids contributing to them because they're all alveolar cell types. And we do find that, as expected, our PNEC expansion media cells are the only ones contributing to the PNEC clusters, for example. So with that, I'll summarize this part. What we've shown is that we've established culture conditions that enrich for normal human PNECs and fetal airway organoids. Our PNECs are maintained in these airway organoids long-term over multiple passages. We call this PNEC expansion media. Using a cocktail of small molecules, we can further promote PNEC differentiation in our PNEC-enriched fetal airway organoids to achieve high numbers of differentiated PNECs. We call this our PNEC cocktail. And then we are currently performing further single-cell RNA sequencing of our fetal airway organoids after addition of our PNEC cocktail to try to identify the PNEC progenitor cell, but also to identify the transcriptional changes that are associated with this path of differentiation towards the PNEC cell fate. So with that, I'd like to thank you all for your attention. I'd like to thank Hans, of course, for all of his support, everyone in the lab. Lisana Den Hartig is a very talented technician that's been working with me. I also have to give a big shout out to all of the clinicians who have worked with me on this, and all the patients who have donated their tissue for these experiments, as well as my collaborators in Jason Spence's lab, Cole Trapnell's lab, and at the International Agency for Research on Cancer. That I hand it over to you. Thank you.